This presentation is about my first year doing my PhD in entomology here at the KZN Museum. I started almost exactly 1 year ago, so I think now is an appropriate time. It was delayed a bit, but it's perfect. So, thank you. So, the very interesting logo you see here is is this two vermilionid flies? I don't know anyone here who knows, but this is the logo of the Diversity of Pollinating Diptera project, which are the people that are funding my research through the museum and allowing me to be here today and every other day. And also, I'm technically a student at University of Pretoria. They'll give me my degree at the end of this. So, who am I? You guys know my name, but it's not necessarily everyone knows exactly what I do here. So, I am, in the broadest sense, a biologist. I study living things. But within biology, I'm within a very particular branch called taxonomy, which has to do with classifying living things. When you find a new species, it needs to be described. How will people tell that this is different from all of the other species? How do we know to recognize it? And that can get pretty technical. You need to give them names. You need to possibly revise old descriptions that are no longer useful, because maybe we have discovered more about those species, and maybe they're not exactly what we thought they were. And the very particular kind of organism that I work with, the kind of biologist that I am, is an entomologist. And that is someone who studies insects. And to become even more specific, because I'm very specialized, I am a dipterist. I am a kind of entomologist who focuses on the two-winged insects, which are the flies. Oh, that's a nice picture of me at the Bloom Museum. Flies. Flies rule, guys. I think no one in the world, it's true, no one in the world is entirely sure just how many kinds of flies there are. They are not just the house flies that you see around garbage. The diversity is pretty dizzying. The number of things that flies do in ecosystems, it's hard to find a role that they don't fulfill in our natural world. You go and look closely, you will see so many different forms. It's quite astounding. You've got musket flies like house flies, you've got bee flies, you've got crane flies, fruit flies, the very weird bat louse flies that live in the fur of bats and eat whatever gets stuck to it.
I'm so sorry.
Hey, go ahead, it's fine. We're just talking about flies. And then fungus gnats, of which there are so many different kinds, and they seem kind of boring, but actually they're fulfilling quite important roles in our ecosystem because fungus grows on everything that decays, and they eat anything made of fungus. Come on in, people. Oh, and that cute little one in the corner there who's keeping the page number for us is a drosophilid fruit fly. So, why do we need to care about flies? Why are there people like me in the first place who get paid to study them? What's the point? Does it actually matter to anyone? It actually does. So like I mentioned, we now know from some genetic studies that there are probably more species of flies, these guys, than any other terrestrial macroorganism. So anything you can see with the naked eye that lives outside of the ocean, there's more species of that than anything else. And they do important things for us, like they actually give us food to eat. Many luxury crops, even the coffee, here's my coffee. The coffee that I'm drinking today in some parts of the world, in some seasons, is actually produced by flies because they pollinate the coffee flowers and produce the coffee beans. Services like that is what I mean. They contribute to our economy, even though most people don't notice them. They also can save us from alien invasions. So I come from a background working in biological control at Rose University in the Eastern Cape. And there we were investigating certain species of flies. And this has been being done for decades, to use them as biological agents to control pests of crops, to control plants that are weeds in many situations, both in like both in the crop field and in wild habitats. And yes, some flies do spread disease. Some of these diseases are pretty nasty. And I had, during the course of my fieldwork, had to take preventatives against diseases like malaria, which is spread by flies. It's one of the deadliest diseases in the world. But that said, some flies are actually being investigated as a source of medicines that can cure diseases. So it's not like flies have only one face and that they do only one thing for us or to us. And these are my babies. So the whole reason I'm here is to study this particular kind of fly, which is something called Rhyncomya. It's a kind of nose fly. You can see he has a cute little nose there. It's a special kind of blowfly. So I'm sure you guys are familiar with those metallic green and blue flies, the blue bottles and green bottles that fly around garbage. Okay, so they're related to them. Besides the basic structure of their body and how they fly, most of what they do is completely different to those flies. The adults are actually associated with flowers, especially in wild habitats. Like really, you mostly only get them in basically untouched wilderness, where they will go and visit flowers of indigenous plants, but also in adjacent agricultural areas, they will visit those plants too. So it's thought that by preserving wild habitats and these guys, they're partly contributing to our crop production. But that's still something we need to learn more about. So it's thought that at least in some places, they're playing a role in giving us mangoes, avocados, coffee. The avocados is mostly in South Africa. Coffee maybe is going on in Ethiopia and Kenya. And the mango is mostly in West Africa, where different species are associated with these crops. And what's even weirder about them is that the larvae don't actually feed on rotting stuff like most flies of this type. They're a very odd kind of social parasite. They live inside termite mounds, pretending to be young queen termites, and we don't know what they eat. There's some evidence that the termites feed them, and then they pop out of the termite mound as these things. So much we don't know about them. They're so unusual. So it's actually worth studying them and learning more about them, especially if they're giving us a possible ecosystem service. And with the project that I'm on, it's diversity of pollinating diptera in the Afrotropical region. So we live in the Afrotropical region, which is basically everything below that red line. It's not entirely accurate. I mean, also there's parts of Yemen that are considered to be in the Afrotropical region, but I digress. All of those colored dots mean something with regards to my work. The blue stuff, all the blue dots, are places where I've received material. So even though I never travel outside of South Africa, I do work on fly material, mostly nose flies, coming from all of those different countries in blue. Ones in yellow are ones where I would like to see material but haven't gotten it yet. And then down here in South Africa is where I actually do my active work and I go out and get that material. So ones in green are places where in my opinion, I've basically fully sampled them. Ones in red are places I should sample but haven't sampled yet. Orange, incompletely sampled. And blue, again, is places where people have collected for me and given me material to work on. So even though I'm here, I am working on things that cover basically all of sub-Saharan Africa, at least with regards to this particular kind of insect. And what I'm doing specifically is working on the Rhyncomya and those roughly 50 species that we think occur in this region. My job is to find out how they fit together and how to properly identify them, because right now we have a situation where most people, possibly not anyone, do not know how to identify and tell some of these species apart, especially in cases where some of the species have a bit of an uncertain status, where it's like different specimens that are supposedly from different species can be very easily confused with each other. And that situation hasn't been fixed because most of the studies were done at a time we didn't have access to DNA technology. So we couldn't define them using their molecules, using their DNA, but now that's possible. And that's one of the main focuses of my project, is to take the way they look, the way that their morphology seems to show that they relate to each other, and see how that matches up with a phylogenetic tree. So that's a branching diagram that shows how these different species actually relate to each other on the basis of molecules, on the basis of DNA. Those are my tools. That's what's available to me. But in order to make this work, I need to examine lots of specimens, and a good chunk of these specimens I need to be fresh, and I need to be able to extract their DNA and analyze them that way. So that's not possible unless I go into the field and collect them myself, in most cases. Fieldwork. It's fun. It's hard. It involves a lot of driving. But while I'm out in the field, enjoying the beautiful scenery, sometimes suffering, I'm working to collect the flies that I'm interested in. So what you're seeing at the bottom there, those two images with little yellow devices in them are different kinds of traps that I can use to catch my flies. So some of them, despite being attracted to flowers, can supposedly also be caught in this kind of red top trap that has some kind of rotting something inside the jar, and the flies will actually fly up to the yellow part and they'll crawl under the red part and they'll fall in and be caught. But I have the best time catching my flies with the very simple yellow pan trap, which is really just a yellow plastic tray that looks to the fly, unsuspecting, like a really big yellow flower. And they love the color yellow. And I just fill it with yellow dish soap, well, water with some dish soap in it, and they just like land in it and they can't get out. Because when you put soap in water, flies can't really swim in it anymore and they just sink. And I mentioned that they live in termite mounds, so sometimes I do actually go and put my back into it and dig up termite mounds in search of the larvae. And those are some of the methods that I employ to get my flies, but most of the time I'm running around like a madman, staring at flowers and trying to like swipe them into my net. And sometimes I actually get cute things like that. And having done that at the end of a field trip, which might take one or two weeks, I have a box full of material like this, where my preference is to just put labels on everything that shows where it comes from, and I need to provide other information like, in addition to the coordinates, so exactly where it can be found on a map, what time I found it, what flower I found them on, other information like if I know the temperature, because that all becomes useful later in working out aspects of their biology, which we really know nothing about. And then some other things happen. Yes. So I have to actually, with a specimen, you don't just get it, put a label on it, and then stick it in the museum somewhere. You need to actually apply your expertise and work out what it is. And that is what a large part of my training as a dipterist goes into. Being able to tell you what that is. I think the animations will work. Yeah, so I need to look at all those different features like the antennae and the wings and the bristles to be able to put a species identification on it. I'm referring to somebody else's description because somebody else went and looked at this and figured out what are the characters that allow you to tell that this is Aldebaria malgashi. And once you have that information, you can then apply other information like, oh, so based on the flower that I found this on, I know that this might actually be a pollinator of the wild pea, for example. And that's how you put the information together. So fieldwork, collecting all those flies and being able to identify all those flies, the actual process of running around can actually be kind of fun. So I get to go to some very pretty places. Like this is Groenbos in, where is it? In Transvaal. And George came along for this one. And I got to see their very lovely entomological lab because Groenbos is a private nature reserve and they actually have some in-house entomologists working for them, which was quite a pleasure to meet them, as well as to see all the pretty flowers and try to find flies. I didn't actually find any flies yet. Besides the beauty of the places that I get to go and work in, get to see wild animals, I get to see beautiful flowers and rugged terrain. It is sometimes very challenging, not just because my tent gets wet, but because these places can be very remote. It can take a lot of time driving around to actually find the place that you're looking for. You can get lost because when you study insects, you tend to work in the hot seasons when you're more likely to find them, when the insects are more likely to be flying. There's a risk of heat exposure. There's a risk of dehydration. Once I almost passed out from dehydration when I was working in the Eastern Cape this year. You get thorns in your feet. You can get injured. Hiking through this rough terrain can chew through your shoes. I had to tape my shoes back together more than once. That kind of stuff does happen. And I don't always have to do it alone either, which makes it a little bit easier. So Cass was with me for some of these field trips. There's Cass catching bee flies because she specializes in bomboliers. This was a lovely place. This was the Knaasvlakte in the Western Cape, which is right on the border. Well, it's where the ecosystem is starting to transition to pure Kalahari desert. This is the succulent karoo, which has very unique vegetation, like these stone plants, which I got to see for the first time, which are little succulents that kind of pretend to be rocks so that they don't get eaten by herbivores. I don't know how effective the disguise actually is, but they can produce some beautiful flowers at the right time of the year. And also they basically don't have any leaves, so they lose very little moisture. These things stay nice and squishy all year long. Very cool. And that's the little house that we were staying in. Also, there's tons of daisies. And some of the field trips I have to do by myself, and these were some pictures from when I was going to Limpopo, which was only a couple of weeks ago, which was my first time ever in Limpopo. And I had to take that medicine so that I didn't get malaria, because I did go to the Kruger. And it doesn't make you feel very good, it actually makes you feel very tired. But that is to stop myself from getting little things in me from the mosquitoes that inevitably bite you. On the way there, I stopped at the Swayim crater in Pretoria, like northern Pretoria, next to Hammanskraal. That is one of the world's best preserved meteorite craters. It's a very important piece of natural history. It's got a unique little ecosystem inside the crater where I did find some of the flies I was looking for. But it's also a breeding ground for mosquitoes. Not the kind that spread malaria, but the kind that bite you on every little bit of exposed skin. When I got back from doing my collection there, the staff looked at me and they were like, they looked at my face and they were like, yeah, those mosquitoes, eh? I was swollen. They bit me on the lips. They have no sense of what's proper. Like, I prefer the mosquitoes here to the mosquitoes there. That was the worst mosquito experience I've ever had. But then when I did finally get to Limpopo, I was collecting in the mostly Mopani bushveld. I saw a baobab tree for the first time. I got to hug one. I saw the Mabalal River, which is a tributary of the Limpopo River that divides us from countries to our north and east. Beautiful place. I did find some flies. A lot thanks to the help of that guy over there staring into the pan with his son. That is William Mosima, who was born at the Mabulela Game Reserve and has worked there almost his whole life. He knows everything about the bush. He's a human GPS. He knows exactly which plants are edible and never had to bring a lunch into the field because he was constantly just like, you can eat this, you can eat that. And somewhere in the middle of Fieldwork, he'd just like hand me a whole bunch of berries and be like, you can eat these. I'm like, thank you. And he actually pointed out quite a good number of fly species that I caught. And I gave some of them as a present to his son, who is also named William Mosima. And while I was there, I learned some other interesting entomological information from the people who live there. So on the reserve, the people who work on the farms also raise these tiny bees, which are called sweat bees or helictids. And they do visit flowers and collect pollen and nectar like, ordinary bees do, but these little guys will also drink your sweat, which you will always be covered in the summer, I guarantee you. And they will use that to make this beautiful clear honey. And that's what they're doing inside those boxes. They'll only nest in dead wood, and you can see that red arrow pointing to one of their tiny faces. They will all come in and out through this one tiny wax tunnel that sticks out of the wood. And they get them to live in the boxes by just opening up a tree, catching the queen, and locking her inside. And then her workers will make a tunnel out for her, but they'll stay in the box. And you can open it up to get honey when it's ready. Beautiful. They're very cute. So I've got to bring my net for reference, but my net's about that high. I saw some of the hugest termite mounds I've ever seen. I was very interested in termites, because they're associated with my flies, and I used to study termites specifically just to know about their ecology. That was cool. Yes, I did see elephants in the Kruger, but in my opinion, the insect stuff was more interesting. And one of the other privileges that you may get as a postgraduate student is the opportunity to sometimes travel the world, which I did early on last year. So, is that? Yes, that's the right one. Okay. The Africa Museum, which is formerly known as the Royal Museum for Central Africa, located in Belgium. These are the funders, most directly, these are the funders of the dipterist project. These are the people who are paying me to work here. And I got to go there, to Belgium, to work with the people at the museum and to work with the material that they had there. I'm one of 2 PhD students that are being funded for this project. So remember, it's not diversity of pollinating diptera in South Africa, it's diversity of pollinating diptera in the Afrotropical region. So the 2 PhDs being funded are me here in South Africa and Tanwet Lamini, who's from Eswatini. And that's a picture of us outside Brussels Central Station when we went on a little bit of an excursion after arriving. And I got to experience cool things about Europe for the first time. This was the first time I'd ever left South Africa. And I love their public transport system. I love that it works. I love that it's clean. That impressed me. And of course, they have cool things like they just have amazing architecture, beautiful cobbled streets with shops catering to tourists. I mean, they are the same shops every 10 steps you take. They just repeat themselves. But it's a lovely place. It's a nice place to. It was a nice place to experience because it's so different from anything that I've ever seen before. And of course, Belgium is famous for having the best chocolate in the world, and I did try some. Now, the Belgians are also strangely obsessed with this statue of a little peeing boy. But we can forgive them for that because most other things about their country are really great. And I thought this was a nice time to talk a little bit about chocolate, because Belgian chocolate is so great. Chocolate, wherever it comes from, comes probably, as you know, from the cocoa that you get from grinding up cocoa beans. But where does a cocoa bean come from? Well, they grow in tropical regions, which includes parts of Africa. And those cocoa beans come from cocoa flowers, which can only become cocoa beans if they get pollinated by an insect. And these are teeny tiny little flowers. So, in fact, the only insect that can really pollinate them are these teeny tiny flies. These 2 kinds of flies are responsible for all the chocolate that you eat, except when the cocoa is hand-pollinated, which sometimes people have to do because there's not enough flies, because they're declining. Let that sink in a bit. So if you see anything like that, you've probably never seen them before. They're so tiny, but they're everywhere. You like these guys, they are your friends. And obviously I was distracted in Belgium. Obviously I was looking at insects when I was in Belgium. They have bumblebees. I've never seen a bumblebee before. We don't have bumblebees in sub-Saharan Africa, I'm pretty sure. No, we don't. So I saw one for the first time and I was like, wow, I've got to get a picture of that. I found that fly that you see circled there. I found this very cool looking fly in an outdoor urinal that was so old it had developed its own ecosystem. I thought that was very interesting. Anyway, I was actually there not to wander around outside, but to work in an office like this one at the museum, which is very big and very old and surprisingly empty. But they have a collection of some of the oldest and most complete collections of flies collected in Africa during the colonial period when Belgium administered most of the Congo as it is today. And some of these specimens are almost 100 years old, some of them are older, and this was my only chance to see them in person and actually have a look at all the details that make up these specimens, some of which define an entire species. So in taxonomy, it can be that a single specimen, which is the type specimen, is considered like, this is your reference, this is the shining example of what this species is. It's based on that one specimen. And if you want to revise a species, to change the definition, update the definition, you have to see these. At least a very good photo, or you need to see them in person, ideally. And that was what I was there to do, to write down information about everything they had, sorted, identified if some things were misidentified. I did have to do that. Update their records and so on. After this whole year, what have I actually achieved, having gone to all these places and done all these things? I attended a really helpful fly identification training workshop in the Western Cape, so I've gotten better at doing what I do. I've examined hundreds of museum specimens, not just in Belgium, but from museums around South Africa. So in addition to fieldwork, I've been going to museums to see what we have. Some of it is on loan now at the KZN Museum, so it's sitting in boxes at my office. Also, the KZN Museum itself has one of the continent's best collections of flies, so it's natural that I should be here. That actually is the reason I'm here. I described 2 new species, I think, and there was supposed to be a picture of one of them there, but now it's gone. But anyway, it was a really nice picture. I'll show you later. As part of a revision of that entire species group, and I've not even really gotten started on my Rhyncomya yet. These guys are just related. More to the point of the Rhyncomya stuff, I've translated dozens of manuscripts, which are the descriptions of all of the Rhyncomya species, most of which are not in English. They're in French, which I don't speak very well. They're in Italian and Latin. So I had to translate them because Google Translate doesn't do entomological terminology very well, and I preferred to just do it myself. So old-fashioned way, with a dictionary. Yeah, it was work. And from all the field work, I think I've collected about half of the species that I need to do the genetic analysis that goes into my thesis. So about writing the thesis, it's also work. I have to read lots and lots of books, lots and lots of old texts, normally in like flimsy reprint from the library, and the librarians were really helpful, helping me get access to these piles and piles of ancient decaying papers that I have to read, and I'm still not through reading them. When I'm examining insects, it's mostly through a microscope. It does hurt your eyes, staring at hundreds of insects, one after the other, squinting like that. And I mostly work on my computer when I'm writing stuff and doing analyses, so sometimes that's me late into the night. And you must take care of yourself. I don't yet actually need eye drops, but I imagine I soon will. I love coffee. I drink a lot of coffee. That keeps me going. And that's actually about it.